Welcome students. Today we are going to discuss about barley and oats. Barley is a member of the grass family. It is a self-pollinating diploid species with 14 chromosomes. The wild ancestor of domesticated barley, that is Hordeum vulgare, subspecies spontaneum, is abundant in grasslands and woodlands throughout the fertile crescent area of Western Asia and Northeast Africa, and is abundant in disturbed habitats, roadsides and orchards. The cereal is not grown in humid, tropical and semi-tropical regions. Outside this region, the wild barley is less common and is usually found in disturbed habitats. However, in a study of genome wild diversity markers, Tibet was found to be an additional center of domestication of cultivated barley. Barley is the world's fourth most important cereal crop in terms of both quantity produced as well as area of cultivation, after wheat, maize and rice. Barley probably came into cultivation about 10,000 years ago. Barleys are annual grasses that may be autumn or spring sown. The Old English word for barley was bear. Grain structure. Barley grains are roughly cigar-shaped, larger and more pointed than wheat, with a shallow furrow running along the ventral side. The kernel of the covered barley consists of the caryopsis and the flowering glumes. The husks consist of two membranous sheaths that completely enclose the caryopsis. Cross cells are in a double layer and their walls do not appear beaded. Only one cellular testa is present, 2 to 4 aileron layers are present, cells being smaller. The main type of cultivated covered barley, depending on the arrangement of grains in the air, are 2-rowed and 6-rowed. The axis of the barley ear has nodes throughout its length. The nodes alternate from side to side. In the 6-rowed types of barley, 3 kernels develop on each node, 1 central and 2 lateral. In the 2-rowed barleys, the lateral kernels are sterile and only the central kernel develops. The color of the grain in covered barleys depend on the color of the caryopsis and the second husk, that is the pally. Color in the caryopsis is due to the anthocyanin pigments or to a black melanin-like compound. Anthocyanin, when present, is red in the pericarp and blue in the aileron layer. The husk constitutes about 11% of the grain. The proportion varies according to the type, variety, grain size, and climatic conditions. Large kernels have fewer husks than small kernels. The husk in 2-rowed barleys is generally lower than in 6-rowed barleys. Chemical composition of barley. The barley grain is rich in starch and sugars, relatively poor in protein and very low in fat. The husk is mostly composed of lignin, pentasins, mannan, uronic acid, hemicellulose and cellulose fibers. The starchy endosperm is composed of about 85 to 89% starch enclosed in cell walls. Embryo consists of about 7% cellulose, 14 to 17% lipids, 14 to 15% sucrose and 34% proteins. Barley grains contain many other substances in smaller amounts, including a range of vitamins, minerals, and phenolic materials. Typically, in plum two-rowed barleys, the husk and the pericarp together made up 9 to 14% of the grain's dry weight. The testa plus the nuclear cuticle plus the pigment strand 1% to 3%. The embryo 2% to 5%, the aileron layer 8% to 15%, and the starchy endosperm around 75%. Pigments in barley. Anthocyanins of the cyanidin type can be found in the green organs of the barley cultivars. Catechins are located in the seed coat and mature kernel. Tannin complexes with protein and often account for off colors that are noted especially during processing. Health benefits of barley. Barley is rich in dietary fiber, which gives food to the friendly bacteria present in the large intestines. These bacteria ferment the insoluble fiber content of the barley to form butyric acid that functions as the main fuel for intestinal cells. Besides this, it is the functional in maintaining a healthy colon. The dietary fiber in barley aids the production of acetic acids and propionic acids, which serve as the fuel for liver and muscle cells. Propionic acid also restricts the action of HMG-CoA reductase, which is involved in cholesterol production in the liver. In this way, barley helps in reducing the cholesterol level in blood. The friendly bacteria that are fed by barley's insoluble fiber also help in removing the pathogenic bacteria from the body. Barley is rich in niacin, which is highly effective in reducing the risk of cardiovascular diseases. It prevents the oxidation of LDL by free radicals. Niacin also lowers the assembling of platelets, which can lead to the clotting of blood. The cereal is effective against diseases like atherosclerosis, diabetes, insulin resistance, and ischemic stroke. Barley's rich content of insoluble fiber helps women against the formation of gallstones. It's also rich in phosphorus, which has an integral part in the formation of body cell structure. Phosphorus is required for the formation of mineral matrix of the bones. Barley helps in maintaining a healthy intestines. It helps in decreasing the transit time of fecal stuffs and also increases the bulk. As a result, the risk of colon cancer and hemorrhoids get reduced to a considerable extent. The cereal contains copper, which reduces the symptoms of rheumatoid arthritis. Copper is also required for the activity of lysyl oxidase, an enzyme required for the cross-linking of collagen and elastin. These substances provide the basic substance and flexibility to bones, joints and blood vessels. Now the food uses of barley. The major use of barley is as a component of animal feeds. It's also used in some foods for humans. Barley may be roasted and ground, the product being extracted with hot water to provide the basis of hot drinks used worldwide as caffeine-free health drink. Malt flour is a source of alpha amylase and other enzymes, so used in baking. Barley malts are also used in the preparation of malted drinks, beers, whiskies, malt extracts and malt vinegar. Oats, Avena sativa, belonging to the family Gramineaceae, commonly known as oat, groats, haber. The word avena is derived from the Sanskrit word avi, meaning sheep, or avasa, meaning foodstuff. Oats are haploid crop which evolved from 3 distinct diploid progenitor species through natural interspecific hybridization and polyploidization. An important characteristic which distinguishes Avena sativa from many wild oat species is the Avena sativa does not shed its grain on ripening. Oats are temperate crop and compared with other cereals grow well in damp, marginal upland areas. Oats are grown throughout the temperate climate regions of the northern and southern hemisphere. Depending on the variety, oats may be sown in autumn or in spring. Avena sativa is an annual grass about 1.5 meters high, clumps tufted or solitary, erect or bent at the base. The leaves are non-articulate green. The inflorescence is diffuse panicle. The grain is tightly enclosed by the hard lemma and palli. Now the production of oats. Oat is produced in various countries around the world. According to the UN Food and Agriculture Organization, the total world production of oats in the year 2008 was around 25.8 million metric tons. Russia being the top producer, followed by Canada, United States, Poland, Finland, Australia, Ukraine. Structure of oats. The common varieties of oats have the fruit, that is caryopsis, enveloped by a hull composed of certain floral envelopes. Naked or hull-less oat varieties are not extensively grown. The hull normally makes up approximately 30% of the grain. Oat kernels obtained by removing hulls are called groats. The groat is considered the whole grain of oats. It is elongated, spindle-shaped, up to 0.5 inch in length and 0.125 inch or less in width. It is generally covered with fine, silky hairs and includes the seed coat layers of cells, starchy endosperm and the embryo. In cross-section, the nucellar epidermis can be seen as a thin, colorless membrane. In endosperm, there is a single allelon layer. The cell walls are not thick. Starch granules are polydelphous. That is, it consists of many tiny granulae which fit together to form a spherical structure. Endosperm cells of the oats have relatively high oil content. Chemical composition of oats. The various components in oats are not uniformly distributed in the kernel. The hull and bran are high in cellulose, pantosans and ash. The germ is high in oil and rich in proteins, sugars and generally ash. The endosperm contains the starch, has low protein content than the germ. Oat groats have relatively good amino acid balance, high in globulin, that is salt soluble fraction, and low in prolamins, that is alcohol soluble fraction. Oats also contain significant amount of dietary fiber, and this soluble fiber, which is composed mainly of beta-glucan and is known as oat gum. Oats also contribute significant amounts of dietary minerals like magnesium, phosphorus, iron, copper and zinc, and vitamins, thiamine, vitamin A, folate and niacin. Avenanthromide in oats. Avenanthramides are phenolic compounds and consists of anthranoic and hydroanthranoic acid linked to one of several hydrocinamic acids through an amide bond. Oats is the only source of avenanthramides. The three most predominant avenanthramides in oats are avenanthamide 1, 3 and 4. Its levels range from 40 to 132 microgram per gram in the oat grain. It is heat stable during processing. Now the therapeutic effects of oats. First is plasma cholesterol lowering. Dietary oats have been seen in several studies to lower elevated plasma cholesterol and in particular low density lipoprotein cholesterol, which is a major risk factor for heart diseases. Modulation of plasma glucose levels. Reduction in postprandial blood glucose responses and associated decrease in insulin levels are important in the control of diabetes. The postprandial blood glucose response after consumption of oat products is low compared with many other foodstuffs. Improving gastrointestinal function. Foods high in dietary fiber exert a number of potentially beneficial effects on the gastrointestinal tract, including decreasing transit time and increasing fecal bulk. Oats and celiac diseases. Celiac disease is characterized by a severe adverse immunological reaction of the intestinal mucosa to gliadins. Gliadins are a component of the gluten protein found in wheat, rye and barley, and these cereals are excluded from the diet of patients with celiac disease. Recent 2 studies have shown that the consumption of oat products did not result in adverse effect in patients with celiac disease. Industrial uses of oats and barley. Oat hulls have also been used as a raw material for fermentation to furfural. Furfural is a chemical solvent used in refining minerals and for making resin. Oats have been used in the range of cosmetic products and cleansing agents, including soaps and facial masks. Various dry and wet milling processes have been developed for the separation of oats into a number of fractions, including oil, gum, starch, and protein. Oat starch could also replace other starch sources in a wide range of applications. Oat protein isolates, including deamidated and acylated oat protein, have some interesting functional properties of use in food systems. These include high viscosity, good water and fat binding capacities, and good emulsifying power. Besides food use of starch, it is also used in the paper industry. Starch is an excellent binder and is used in the manufacture of charcoal, ceramics, gypsum board, and chalk. Till now, we have discussed about the structure, chemical composition, industrial uses, and therapeutic effects of both barley and oats. Hope you have understood. Thank you.